leave enough time for the next speaker, who's a good friend. So I'd like to welcome you to this session. I'm Justin Starren. I'm wearing a number of hats here. One as the Chief of Health and Biomedical Informatics at Northwestern. I'd like to say welcome to Chicago. And I'd also, Mike Bisich asked me to plug the fact that this meeting is really a warm-up for a lot of informatics that's happening this month in Chicago. We actually have a mayoral proclamation declaring Informatics Week in Chicago from October 30 through November 7. And yes, it's more than a week. We couldn't fit everything in. We'll have a number of lectures and activities about informatics leading up to the AMIA meeting. In addition to all of the normal AMIA activities, we'll also be highlighting informatics activities in Chicago. So those of you who can, I know travel budgets are light. It's looking like an absolutely fantastic AMIA meeting. Registrations are already above prediction for the meeting, so we're expecting an absolutely jam-packed meeting, a number of late-breaking sessions that should be of interest to folks, so I encourage you to come to that. I also wear the hat as the co-chair of the eMERGE EHRI integration work group, which is really what's driving this talk. And for those of you who find your email a more interesting thing to do than listen to me, the top article in CAP today is all about next-gen sequencing and clinical application. So if you miss something here, you can probably find it in that article. Now, I'm curious, how many of you right now are doing CLIA-grade sequencing in your operations? Okay, so we've got a few. So something for you to read on the plane home, released yesterday by the Presidential Bioethics Committee, was a big fat report about the issue of privacy and next-gen sequencing, and this has been picked up vigorously by the press. Everybody's got it. This is Washington Post, CBS, Fox, you name it. It's out there, so you can download the PDF, read it on the plane, because you'll probably get questions about it by the time you get back. This is what it looks like, and this actually sets a good theme for this talk. This is a quote from the report itself, that we are in a period of intense transition with respect to integrating whole genome sequencing into clinical care, which is exactly true. We are at the inflection point, which means the answers to most of these questions are changing almost on a weekly basis. Everybody has probably seen this graph about the drop in the cost of sequencing, right? The estimate is this year we are down to about $3,000 for a whole genome sequence, which to benchmark it is the same cost as a BRCA panel. So the era of doing 1 or 2 or 5 genes is coming to an end very rapidly. So just to make sure everybody's on the same Omic page, right? The genome is all of the genetic complement. A phenome is the observable output of that. So a phenotype, right? A SNP is a single nucleotide polymorphism or one base change. A haplotype is really a statistical observation. It's a whole bunch of SNPs that tend to co-migrate. We talk about alleles, which are alternate forms of a gene. GWAS, a genome-wide association study, is what we've been doing with the Illumina chips, where you will have a number of sequences that detect SNPs, up to 1 million or 2 on a chip now. And the new abbreviation you're going to see a lot, WGS, whole genome sequencing. This seems to be overtaking NGS, or next-gen sequencing, as the operative acronym. And you can have an enneome, right? We have the genome, we have the proteome, we have the metabolome, we have the epigenome. If you want to talk about exposures, we're now talking about the exposome. So ohm has now become sort of the synonym for stuff, and you can put almost any word in front of it. The eMERGE network is a network funded by NHGRI. It stands for Electronic Medical Records and Genomics. We are now just going into year 7 of the group. We started as 5 sites. We are now up to 10. And the goal is to do a whole bunch of things. We started out on the research side, saying, can we use the data that's in electronic health records to do decent genomic science? And in all the discussions of big data and pooling data and HIE, that sounds like a well-duh kind of a notion. But you go back 7 years, there are a lot of people who actually believed it was impossible. We also spent a lot of our work dealing with the issue of how do research subjects and patients feel about this. So issues of consent, of access control, of privacy. In the new phase, we are also getting into the other end of the pipe, taking findings and trying to bring them back into the electronic medical record, which is what I'm going to spend most of my time talking about. So the original 5 sites, in order to get into the club, you had to have a genetic biorepository and you had to have it linked to an EHR. So the 5 founding sites were Group Health, the Marshfield Clinic in Wisconsin, Mayo Clinic, Northwestern University, and Vanderbilt. And this graph gives you a sense of the relative biobanks. So Marshfield actually has the oldest large-scale genetic biobank in the country. Theirs was 30 years old when we started, 35 years old now, in terms of the amount of EHR data that they had for each of their samples. Mayo holds a record for the oldest piece of digital data in their data warehouse at 106 years now. And Vanderbilt blows everybody else away in terms of raw size. They started out at about 80K. They are now over 150,000 samples. Different group of EHRs. Some of the biobanks started as disease-specific, some of them were geographic, some of them were population-based, and some of them were collected from leftover blood that went through the regular clinical lab. The questions we asked in the first round were technical. Can you even get data out of an EHR in a way that you can compare it across sites? And what are the ethical, legal, and social issues? Given the amount of time that we have, I won't go into the detail of the eMERGE Phase 1 findings. Short answer is yes. You can do reliable phenotyping across multiple institutions. Don't expect to do it without natural language processing. There is still far too much data in the text notes to get the nuance that you need. Josh Denny at Vanderbilt decided to say, well, let's turn this whole thing on its head. Right? A typical genome-wide association study, you take one particular attribute or phenotype, and then you look at a whole bunch of genes and see which one seems to be associated. He said, let's take one SNP and let's run it back through the data warehouse and say, are there clinical findings which are unusually common in people who have alterations in this particular gene? So this is an example of one of them. I'm not even going to read the RS or reference sequence number, but it's a gene associated with hair color. And what you see is a number of diseases that are not surprising having a gene that's associated with hair or eye color. So skin cancer, basal cell carcinoma, actinic keratosis, neoplasms of the skin. So In some ways, this is a validation of the GWAS findings, that the association goes both directions. Here's another one, hypothyroidism, which is associated with FOXE1. And then if you look at a whole bunch of diseases, the important factor here is all of the bars. Those are 3 different SNPs associated with hypothyroidism and a bunch of different diseases. Everything that is to the right of the dotted line is positive, and everything where the error bars don't reach the dotted line is also statistically significant. So quite a number of statistically significant results. Josh didn't settle for that. He did the many-to-many association, so he took all of the published GWAS associations from the NHGRI panel, fed them back through their data warehouse, and this gives you a sort of graphical sense of how many associations he found. The farther to the right the dots are, the more significant the association. eMERGE 1 has produced, for those of you who are into research, a large number of dB-gap data sets. Many of these are out of embargo. Some of them are coming off embargo soon. We've also produced something called PHEKB, or the Phenome Knowledge Base, and this is the repository of all of the EHR-defined phenotypes that we have developed in a pseudocode version. This is also now our workboard for developing new ones, and it's really easy to get to. It's phekb.org. So if you want to find patients in your databases with a particular phenotype, we've now run into the buzzsaw and fallen on the sword for you, so you can benefit from our pain and suffering. One of the things that we learned doing phenotyping is that finding the controls is a lot harder than finding the cases. And one quote that we repeated more times than I can count, the one from Carl Sagan, absence of evidence is not evidence of absence, that when you're dealing with EHR data, we only ask patients about things we think they might have. We don't ask them about everything. So if you want to be certain that a patient doesn't have a condition, you have to make sure that some test or some survey or some structured instrument was delivered that would have picked it up if it had been there. And that's a critical factor when you're analyzing this data. It's very different from analyzing a standardized randomized control data set where everybody has to give a yes or no answer to every question. We've also developed quite a number of software tools, including a number by Brad Malin focusing on patient privacy. And again, I'm not going into these in detail for the sake of time and also to allow some time for discussions, but www.gwas.org, you can get to the website, get access to the tools. So phase 1, we were trying to use this for research. We were trying to do genome-phenome associations. In phase 2, we've added more sites. We've added pediatric sites. We're trying to do this faster, better, cheaper. And in particular, we're trying to integrate genomic information back into clinical care in a way that can be applied across multiple sites. Again, this was consistent with the theme of eMERGE the first time around. People were getting some data out of one EHR and doing one study with it. The question was this replicable? Could you develop a method that could be applied at more than one site and come back with the same answer? Well, now we're trying to do the same thing with respect to genomic decision support. We've come up with the phrase, to paraphrase some other comments, crossing the Omic chasm. What we've realized is that getting to where we envision in the future, which is a polyomic or pan-omic world where we're measuring lots of omics on our patients and pouring all that data back into the EHR, is going to require a fundamental change in the way we think about this process. We've seen that slide, right? Sequencing is getting cheaper. This is an interesting study that is coming out this year. They looked at patients, primary practice, medical home cohort, and they followed them for 5 years, and they said, how many of these ended up on a gene that we have a measurable pharmacogenomic association with? And what you can see is that 65% of those patients over a 5-year period got at least one medication that had a pharmacogenomic component to its dosing. So this isn't a case anymore of the rare or the unique or the unusual patient. This is a case where genomic information will be part of our care for most of our patients. As part of this second phase, we talked to both patients and clinicians about their perceptions about genomic information and their electronic health records. Overall, patients think it's great, right? We can tell you in advance, what does the doctor say? Take 2 and call me in the morning, right? Well, what if he didn't have to have the call me in the morning because he was much more certain about whether this drug would work for you or not? So they think that's great. They are worried about cost, and they are very afraid of the future hireability, privacy, insurability. They are not comforted by GINA. And it's important to remember that GINA covers some types of health insurance. It doesn't cover all types of insurance, right? It does not cover life insurance. It only covers health insurance. One issue that also comes up is when you do genomic testing on a patient, you discover things that you can infer about their entire family. People who may not have given consent to the testing, you're learning things about, which raises all sorts of legal issues. And if you talk to the folks who maintain these biobanks, what you find is you also find a fair number of cases of non-paternity. And that may or may not be known to anyone involved, and dealing with that can be very dicey. Many patients want the information, not all of them, but they pretty much all agree that they want to be the ones deciding what they get, which also creates ethical conundrums when they say they do not want information that if they don't have may kill them. Talking to the physicians, a number of things became clear. One is physicians are a skeptical lot, right? Every year there's an article in JAMA about this new thing that's going to revolutionize medicine and change the way we do everything and cure cancer and save everybody. And we also know that of those articles in the major journals that are these massive breakthroughs, 2/3 of them turn out to be wrong 5 or 10 years later. So doctors are skeptical, and they are skeptical about genomic testing. They say this is the latest, greatest gee whiz thing, but I'm not really sure. And don't tell me about things that aren't actionable. I've got enough things to deal with that I can deal with. Don't tell me about things that may be true but I can't do anything about. They also feel totally unprepared to deal with this information. It's changing so fast that if you graduated from med school more than 24 months ago, you're out of date. Sometimes I think if you graduated from med school more than 24 hours ago, you're out of date. This is going to change the way we think about electronic health records. Genomic data is not like data that we typically put in our electronic health records. And by electronic health records, I mean the thing you buy from Epic or Cerner or any of the vendors that are getting meaningful use certification for their electronic health record. I realize that many of the large vendors also sell a number of ancillary systems, but I'm talking about the core EHR. Genomic data is huge. It's a lot bigger than a whole slide file, right? We're talking about 3 billion base pairs for a haploid sequence, and that's without any annotation, without any epigenomics, without any conformational or 3D modeling, without any of the other omics overlaid on that. You can't give this to someone in raw form, I don't care how trained they are, and expect them to interpret it. Okay, any more than you could give someone the raw beam trace coming out of a CT scanner and give them a slide rule and expect them to reconstruct the image. I mean, it's that much data and that complex a problem. We are going to be collecting more data than we can use. This is a new way of practicing medicine, right? When we order a test, what do? We write down the use for the test in order to get it paid for. So we have to know what we're going to do with the information before we ask for the information. When we do whole genome sequencing, we have no idea what to do with 99.9% of the stuff that comes back. Our belief is that someday we will know what it means, but we're now collecting data before we know what to do with it. That's a new way of practicing medicine. Most of the genomic data that is out there, including the stuff that is being used for treating patients, is not what I will call really CLIA grade, which is to say, if you have a research lab that is CLIA grade and you develop an assay, you can grandfather your CLIA in, but that's not transportable to any other site. So there is very little fully CLIA grade next-gen sequencing out there. And as I mentioned, the cost is plummeting. So very soon it'll be cheaper to order a whole genome sequence than it will be to run a TACMan on a dozen SNPs. What do we do with our omic data now? And this is our genomic data, and most of our omic data follows the same path, right? Patient comes in with a clinical presentation. Doctor suspects something. They make an order for a genetic test. It typically goes out to a reference lab. A PDF file comes back, or a piece of paper comes back that gets scanned into a PDF in the EHR. Then some sort of a genetic counselor or other genetic specialist reads the paper, writes a consult note that then gets discussed with the primary clinician and the patient. The issue here is this is not computable, okay? The only way this works is by inserting a highly paid and highly trained specialist into the loop. If you talk to folks who are hiring at most large medical centers, they will tell you they're already having trouble filling their slots. There's already a shortage, and the pipeline is not getting bigger anywhere fast enough to capture more than a tiny fraction of the testing we're going to be able to do. So we can't keep the genetic specialist in the loop for every one of these tests going forward unless we're going to have 10 or 15-year turnaround time on a lot of these lab results when we scale up. The other big issue is actionability. So at the highest level of actionability is pharmacogenomics. Your genotype and this gene do not get along together. This is a bad thing, okay? Or a very high level occult disease risk where when you get this genotype back, you should be ordering some sort of phenotypic or physiologic assay to confirm or exclude that disease. The people who deal with that level are the clinicians. If you go down a level, you have a disease risk where we can't actually do a lot about it except tell you to do something different. So the cardiovascular risk profiles are a great example of this, right? We've been telling patients about their Framingham scores for years. Well, some patients changed their behavior, a lot of patients didn't, right? If patients did everything doctors told them, we wouldn't be talking about obesity. On the other hand, we wouldn't have obese doctors either. So, I mean, it's very clear that this isn't exclusively about the patients. There are also risk scores where you have an increased risk of developing a disease. You don't have it. We don't know if you'll get it. You just have more risk than somebody else. In those cases, we have watchful waiting. Those behaviors typically fall on the patient. But even these are a tiny fraction of the data that we get out of a typical GWAS. Most of the GWAS studies and the whole genome sequencing we do, if there's an association at all, it's a small, you have a 5% increased risk of diabetes. Or you have a 3%, statistically significant, but 3% increased risk of early onset Alzheimer's. Well, for those low associations, that's really consumers. That's what we're getting from recreational genomics, right? Which is what people are calling when the patients send their own samples out and get the test back. When you do a whole genome sequence, though, most of the stuff goes in the bottom bucket. Your genotype's different. We have absolutely no idea what it means, except that it's different. And right now, it doesn't do any benefit to the patient or the consumer to tell them about those, because all we will do is make them crazy. As I mentioned, the eMERGE Consortium is about bringing the idea of bringing genomic testing back into the clinical workflow. All of the institutions are going to be doing a pharmacogenomic pilot looking at clopidogrel, Coumadin, and statins, as well as warfarin. And then each of the sites also has some target genes that they will be looking at. Many of them are pharmacogenomic. Some of them are disease risk profiles like Mayo. Some of them are these occult diagnoses where if you are a homozygote, it's worth testing to see whether you actually have the disease, because often by the time you present clinically, it's a lot harder to treat. CYP2D6 is another pharmacogenomic. It's associated with metabolism of opioids. There was an article in the popular press, I think it was about 3 months ago, of a baby dying because with CYP2D6 you can either be a low metabolizer where you don't convert codeine into its active form, and so you might as well be eating M&Ms. In terms of pain control. But there are some folks who are hypermetabolizers, and they convert it extremely quickly. And there was a case of a mother who was getting codeine for postpartum pain, was a hypermetabolizer, put so much of the active compound into her breast milk that the baby had an apneic episode and died. So aside from comfort, this actually has safety implications. In a nutshell, this is how far we've gotten. So most of the sites have selected all of their variants. Most of the sites have done most of their user input selection. And to be fair, the three on the right, the pediatric sites, were added about 8 months after the rest of the sites, so they're playing catch up. Getting internal approval for these types of studies through your IRB can be an interesting process. And it is interestingly interesting because all of these studies are only looking at genes where a national panel has identified a pharmacogenomic correlation and said this should be done. So this is a best practice behavior. Notwithstanding, a lot of IRBs are still extremely leery of these types of studies. Then we have to figure out, okay, so we can collect the samples and we have the consents and we can measure the gene. How the heck do we get that back into our EHR in order to make a decision support rule fire? Then we have to actually write the code that does that and roll it into production and get it out and get the doctors reacting to it. And finally, as you can see, we haven't really started much evaluation yet. See what happens. Now, Vanderbilt, as you can see, is out in front of everybody. They're building off a prior study called PREDICT, which is pharmacogenomic research for enhanced decisions in care and treatment. So they've been doing this for several years now. Their primary target is CYP2C19. And for those of you who don't remember what that does, this is a drug that affects the metabolism of clopidogrel, which is a drug that is given to folks who have stents, and in particular, the folks with the STAR2 homozygous mutation. So those folks right there seem to have an increased risk of clotting when they have drug-eluting stents and are on clopidogrel. So they need to be switched to a different medication. The other medication has higher side effects, which is why we don't put everybody on it. So what they have done at Vanderbilt is they've developed an algorithm to figure out whether people have a risk or a probability of getting stented or ending up on warfarin in the next 3 years. Everybody who is above a certain threshold, they say, hey, can we genotype you for free? And then they store that in the electronic health record. Then if a doctor orders clopidogrel on anybody who's a STAR2 homozygote, they get this screen that says, probably a bad idea, and then gives them, the doctor, an option to either change the medication or put a note in the chart about why they want to proceed with clopidogrel anyway. As the group has discussed how to actually make this work, especially in the era of whole genome sequencing, we've come up with this concept of what we call the Omic funnel. So when we do whole genome sequencing, right, we get a huge amount of data. And right now we're doing whole genome, we're going to be doing whole genome methylation, we're going to be doing tissue arrays, right? We know in cancers that the different clones of the cancer have different genotypes, so we'll be collecting all of that data. Soon we'll be collecting genomic tertiary structures. Most of this is just raw data. It's not something you want to put in an EHR. And in fact, the raw sequence is just a bucket of stuff. So the first thing that has to happen is that software is run on that in order to find attributes, features, variants. Now those could be SNPs, they could be insertions, they could be deletions, they could be rearrangements, they could be copy number variants, they could be methylation of a promoter site, they could be all sorts of things, but they're identifiable features in this person's genome that we can label and then have a list of things about this person that are significant. So we have all of those. But as we said, we have no idea what most of them mean. So what has to happen is that then that list has to be filtered again against those features or attributes which scientific consensus has said you should do something about this. And there is a consortium, the CPIC Consortium, which is developing a national list of clinically actionable variants. So having done these 2 steps of filtering, now we have not just genetic differences, we also have differences that make a difference. In most patients, we still have more of those than a human can easily wade through. So the next step is we have to combine patient-specific data. I think a great example of this is if you look at warfarin dosing. We can explain, warfarin dosing varies over an order of magnitude, but it has an extremely narrow therapeutic range. And I can tell you from personal experience how narrow that therapeutic range is. I take 13 and a half tablets a week. If I take 13 or 14, I am out of range. Okay? The issue is individual patients vary by more than an order of magnitude about what dose keeps them in range. We can explain about 1/3 of the variation between patients based on their genomic profile. Turns out we can explain another 1/3 of it based on their clinical profile. Whether you are getting anticoagulated because you have a device in you, like a heart valve that they don't want to get gummed up, or whether you're being anticoagulated because you have had a thrombotic event, has a significant impact on how much it takes to get you in therapeutic range. So you need both of those, and even then you're only accounting for about 2/3 of the variability. But 2/3 is better than none. We also know that there very quickly will be far too many variants for each hospital to be expected to write its own decision support rules to do this. And so there are several consortia out there, including one by Blackford Milliton, looking at ways to develop a national, a national repository of rules that could be run in EHRs. This database sort of exists now. This database is still mostly a pipe dream. The hope is that if we pull all those things together, we can pour raw sequence in the top of the funnel and get clinical behavior out the bottom. For those of you who are used to the informatics way of thinking, we're essentially going from data to information to knowledge to action. Or if you're in the CTSA world, right, we're going from bench to bedside. That's a good theoretical overview, but what does that mean in practice? If you look across all of the eMERGE sites, what we found is that there are a limited number of common pathways for how this data ends up flowing. So this is the traditional route, right? We have a textual result, it goes into a reports holding area, gets read directly by a human. The HL7 standard does support transmission of certain genetic tests as an HL7 standard. The challenge, of course, is that were you to take an entire gene sequence and all the variants and put them in OBRs and OBXs, you would end up with a multi-gigabyte text file. So for things that you know are actionable, though, there is an HL7 path that can feed through lab results, which then you can run off your traditional EPIC or Cerner or whoever CDS. Typically, though, we're doing whole genome sequencing or we're running GWAS chips. So we're getting a whole bunch of stuff, right? If you run one of the small chips, like an ADME chip, which is a drug metabolism chip, you get 184 variants for 34 genes, of which you're using maybe 3 right now. So you have to have a place to put all the stuff you don't know what to do with. So for complex data, it ends up going into some sort of an Omic repository that sits off to the side of your EHR, but is still behind your clinical CLIA grade firewall, which is an important factor, that you can't just grab the research database and throw it in there, because once you've done, it's tainted and you can no longer use it. It has to stay clean. You then run your analytics. You decide what are your actionable attributes. Maybe it's your CYP2D6 or whatever that you want to pass into the clinical workflow. You have several ways of doing that. One, you can basically create an observation that you pass through to your EHR, which then runs your decision support. Or you can put a hook in your decision support, and this is the hope for many of the national approaches, where when the CDS gets to something that has a genomic factor in it, it doesn't try to answer the question. It sends the relevant data out to an Omic decision support system, and that sends the answer back. Every one of the sites in eMERGE is using one of those paths. So far, we haven't found anybody who's doing anything that doesn't follow any of those options. But there are a huge number of open questions. What goes in this ancillary? What goes in the EHR? At what level do you filter it? How do you decide what goes where? Which of these decision support options is better? Don't know yet. You can't buy that Omic's ancillary yet. You can buy the repository. There are several repositories that you can buy that will hold your Omic data. Oracle sells one, SnpMax sells one, so there are some of those. But the rest of it, the analytics, the decision support, the messaging, that you're going to have to build your own. Format standards and codes. We're making progress, and we'll talk about that, but turns out we don't have a good way of naming genes that scales. We've been naming them for research sort of as they come up, but now that we're doing whole genome sequencing, we're discovering hundreds of variants. Some of the whole genome sequencing studies have suggested that virtually every person they've looked at has about 30 variants that they have never seen before. Now, at some point, that should asymptote because the number of people on the planet exceeds the number of base pairs. But so far, it has not asymptoted. And the incidentaloma. So Predict took a stab at this. They took all of the SNPs that they were measuring on the ADME chip, looked at all of the other diseases and things that they were associated with, and said, okay, what do we have? You know, what are the things we're finding in these people that aren't related to clopidogrel? This is a typical example. So this is a healthy Caucasian female. The genes that they tested, okay, have associations, risk factor changes with, oops, where were we? Bladder cancer, breast cancer, colorectal cancer, gastric cancer, kidney disease, lung cancer, inflammatory bowel disease, and Parkinson's. What do we tell this person? Right? Do we give her all of this information? Right? In some of these cases, these are small risk increases. In some cases, it's telling someone, you know, moving to Australia will increase your risk of being hit by a meteorite by 100%. You know, I think of the 2 people who've been hit by a meteorite. You know, one of them was in Australia and one of them was somewhere else. So yeah, you're probably not going to get hit by a meteorite if you move to Australia, but it will definitely increase your risk. Also, you know, we all learned this in our training, right? DNA makes RNA makes protein makes everything that happens. But now we're finding that there's all sorts of other stuff going on, right? We have epigenetics. We have methylation. So we're actually changing the effective DNA content in real time and passing that on to our siblings. We know that most of the DNA doesn't code for protein. But we also know that one stretch DNA turns out may code for 10 or 20 or 30 different proteins because it gets rearranged and reassembled to make completely different proteins. We have copy number variants, right? So it's not just do you or don't you, it's, well, I've got 3 of this kind and 2 of that kind and 1 of that one and 4 of these. How do we make a risk assessment of that? We don't know yet. And we know that genes affect other genes, right? A huge amount of DNA makes RNA that then binds the DNA that then changes the way the DNA is expressed. We know that a lot of our genes are heterozygous. We're not quite sure what, we're very good at homozygous states. We're not good at inferencing over heterozygous states. CYP2C19, is their heterozygote good? Is the heterozygote bad? Are they high metabolizers? Are they low metabolizers? Are they intermediate metabolizers? That has changed at least 3 times just while I've been in the eMERGE study. And, Watson and Crick were wrong. The 2 strands of DNA actually aren't complementary. They're mostly complementary. But there's 0.5% that misbehaves. It's in loops and assorted other things. So the 2 strands of DNA aren't even identical. So the genomic variation between individuals is a lot more than we thought it was. We also know that heredity, right, we can measure more heritability of many diseases than we can count for with all the known genetic variants. The implication of that is that as we're moving to genomic medicine, family history is not going to go away. It's not going to simply be replaced by gene sequence. Family history is going to continue to be important as a way of dealing with all the stuff we don't know what to do with. We talked about epistasis and the interaction between genes, right? This is one particular gene interaction network, but nowadays we can draw hundreds or thousands of these, where some genes are driver genes and they upregulate some genes and downregulate other genes. It turns out for cancer that a lot of our gene hunting studies were wrong because they were picking up the downstream genes which showed big effect, and it turns out that the changes as we're doing the sequencing are actually in these driver genes. So one area where we are making progress, though, is in the formats. The 1,000 Genomes Project has come up with something called a variant call format, which is becoming really the final common denominator for at least recording sequence data. So the idea here is that each person's sequence gets compared against a theoretical archetypal reference sequence, and then all of the differences are noted in a file. So this is an example where you have a chromosome, you have a chromosome and a position and what the reference sequence is and what the patient has and how many times the sequencer, how many copies the sequencer had of that particular sequence, so it gives you a sense of how confident you are at that call. I mean, it's important to remember that 2 years ago, the best next-gen sequencers we had on the order of 500,000 errors per genome. If you have 3 billion tries, you can be 99.99% right and still be wrong a lot. Also, polyphenotypes, right? Most of our research has looked at the effect of a gene on a disorder. Most people don't have a disorder. They have a whole bunch of disorders. Some of them will be the result of a common gene. Some of them will be a result of gene interaction. So we have a complexity explosion that we are not even remotely equipped to deal with. So what are some of the takeaways? Well, this is definitely coming. It's not here yet, but you can hear hoofbeats. It requires changing the way we think about information, the way we handle information, the way we think about decision-making in healthcare. This is a huge project. This is just a small fraction of the people. Primarily, these are people I stole slides from. As we said, it's funded by the NHGRI. And heretofore, I'd gotten through an entire talk without a New Yorker cartoon, so I had to throw one in to end. This is one of my favorites. And with that, I believe we have 0 minutes for questions. I've already taken some of Paul's time. But I will be around after Paul's talk during the break so you can ask your questions, but I don't want to take any more of his time than I already have. So, thank you very much.
Okay, so where is this?